To maintain physical health, we usually take dietary supplements like vitamin C or protein. Then, what maintains our mental health? 320 million people, which is almost the same as the population of the United States, actually represents the number of people suffering from depression worldwide. Nearly half of all global depression cases are found in Asia, with Japan, unfortunately, leading among the G7 countries in suicide rates. Suicide is the leading cause of death among the productive population of 10 to 30-year-olds in Japan. According to a WHO survey, 98% of suicide victims have a background of mental illness, highlighting the significant issue of mental disorders in Japanese society. Depression is a mental disorder characterized by profound sadness that disrupts daily life, loss of motivation, and interest in things, and it falls under the category of mood disorders, along with bipolar disorder and others. Currently, many individuals are working towards the treatment of depression, primarily utilizing pharmacotherapy centered on SSRIs and SNRIs, as well as psychotherapy, which involves counseling and extended discussions with a therapist. We actually communicated with experts in the field of depression and came to realize an important thing. It is that the awareness of preventing depression and daily self-care for mental health is not yet widespread. Pharmacotherapy and psychotherapy are effective in treating depression, but they are methods employed after the onset of depression and cannot prevent it in advance. Furthermore, due to the strong side effects of medications and the high cost of treatment, they are not suitable as simple self-care for mental health. So, we focused on 3 components found in the flower called saffron and proposed creating an affordable and simple means of supporting mental health. Saffron, which has been produced in high quality in Japan for about 300 years, contains 3 antidepressant components known as crocin, crocettin, and picrocrocin. Clinical studies have confirmed that these components have similar or even greater antidepressant effects compared to anti-anxiety drugs, with significantly fewer side effects. However, obstacles such as high production costs and the difficulty of genetic editing are currently major limitations to widespread use. Therefore, we attempted to produce these 3 antidepressant components using Escherichia coli. By incorporating these bacteria into cookies and offering them to people, we envisioned that it would be possible to provide mental health support simply by consuming cookies, establishing an affordable and effective self-care approach. We named this project Saffricure. But why cookies? Depression patients are advised against foods like bread, cookies, and cake due to their potential to cause blood sugar spikes followed by crashes. However, inadequate carbohydrate intake can lead to low blood sugar and depressive symptoms, so balance is crucial. Hence, we chose oatmeal as the primary ingredient in our cookies because it contains blood sugar-regulating beta-glucans. It is also preferable to include nuts and high cocoa chocolate. Importantly, none of these ingredients inhibit the effects of the compounds we produce. Additionally, they help reduce unnecessary stress by alleviating guilt associated with sugar consumption. Therefore, these cookies could potentially play a crucial role in addressing these issues. We started our research taking these into account. First of all, we attempted to biosynthesize crocin and picrocrocin using the common E. coli strain BL21, without any special substrate addition, and succeeded. Biosynthesis of picrocrocin in vivo by heterologous expression has never been done successfully, and it was an extremely difficult task. However, we have utilized engineering cycle to overcome this difficulty. First, we examined the production of crocettin by introducing parts assembled with PRK404, a duet 1 vector, into DH5-alpha and BL21 strains. Crocettin production was confirmed in both DH5-alpha and BL21 strains by HPLC mass analysis of extracted culture medium. Next, we will discuss the production of picrocrocin. At first, we tried to confirm substance production by introducing parts which we call 00 that have enzymes up to HTCC, and parts which we call 01 that have enzymes from HTCC to picrocrosin into BL21 strains separately. However, this did not work. So parts 01 was assembled downstream of parts 00 assembled in PRK 404 and introduced into the BL21 strain. The reason why the original strategy did not work was that the promoter of parts 00 was an endogenous TAC promoter in E. coli, while the promoter of parts 01 was a T7 promoter. So the enzyme upstream of the biosynthetic pathway was not produced, and only the downstream enzyme was produced. Fortunately, the intermediates in the biosynthetic pathway we designed were carotenoids, so we were able to run the DBTL cycle based on colony color. The supernatant was analyzed by HPLC mass to confirm the production of picrocrosin. Parts 01 is a saffron glycosyltransferase. This was the first successful heterologous biotransformation of picrocrosin from HTCC using the above enzymes and the first biosynthesis of picrocrosin in a heterologous organism under conditions that did not include special substrates. On the other hand, we attempted in vitro conversion of crocetin to crosin using a bacterial extract prepared from a strain expressing UGT94E13 and UGT74F8, known to catalyze this conversion in E. coli. However, we were unable to detect crosin in the reaction mixture. Moving forward, we need to verify the expression of enzymes and explore extraction methods or mass spectrometry detection conditions for crosin. We originally planned to extract the components from E. coli and include them in cookies. However, in the course of our experiments, we realized that the extraction of the target substances required 4 times the amount of methanol and chloroform as culture, and we thought we could produce them at a lower cost by eliminating this process. Therefore, we concluded that we could provide the cookies directly containing E. coli with the 3 antidepressants in vivo as a faux shoe. In that case, the BL21 strains is not suitable for human consumption, as is from a probiotic point of view. Therefore, in line with our goal, we aimed to synthesize a saffron-derived antidepressant component using the E. coli Nissl 1917 strain, which is approved for human consumption by the FDA. We introduced parts 00 assembled in PRK 404 into Nissl 1917 strain. We could not detect crosin, although we could assume that carotenoids were being produced because the colonies and extract were clearly yellowish. Based on these results, we engaged in discussions with depression patients, their families, and experts to prepare for the product's introduction into society. In comparison to competing antidepressants, we confirmed several advantages of our product, including minimal side effects and low psychological barriers associated with its consumption in the form of cookies. And that's not all. We have come to one major decision through these discussions. The Cartagena Act now regulates the growth of genetically engineered organisms. E. coli, which is capable of proliferating, cannot actually be transported to the human mouth under the Cartagena Act, even if it is put in a cookie as is. It has been a major hurdle in social implementation of products. We have repeatedly discussed how to address and handle this situation together with team members, researchers, and companies. Finally, we decided to carry out an additional project in a short notice, to break down the chromosomes of E. coli bacteria that had accumulated antidepressant components, thereby eliminating their ability to multiply. Since the Cartagena Act regulates the growth of genetically modified organisms, we thought that if we could eliminate the ability of E. coli to grow, we could dramatically lower the regulatory hurdle for providing cookies. Because we envisioned social implementation, we sought a system that would minimize the incidence of escapes. It is known that it is possible to create chromosome-free E. coli by using chromosome-selective restriction enzymes and a strict expression control system induced by crystal violet. We proceeded with our project in a manner that emulates this. Finally, we succeeded in confirming that E. coli Nissl 1917 and E. coli BL21 were no longer proliferating by colony formation, and in confirming chromosome-free by fluorescence microscopy using the BL21 strain. We confirmed the absence of colonies on LB agar plates with appropriate antibiotics and CV for both transformed BL21 and E. coli Nissl 1917 strains after overnight incubation at 37 degrees Celsius. However, some colony formation occurred on CV-containing plates, likely due to escape mutants. To further verify the chromosome-free status, we used DAPI and fluorescence microscopy, observing samples at optical density at 600 equals 0.2 after CV induction at 0 hour, half an hour, 1 hour, 2 hour, and 4 hour. Only the CV-induced sample showed loss of fluorescence, indicating chromosome-free status achieved by the parts which we call 0-2. The strength of our system is the possibility of maintaining the functionality of the designed plasmid even when it becomes chromosome-free. We have been performing IPTG induction at optical density at 600 equals 8 when looking at material production in parts 00 and others. We challenged whether we could confirm substance production in chromosome-free E. coli by performing CV induction at the same time. We confirmed that the samples became chromosome-free 30 hours to 72 hours after CV induction. We also confirmed the production of crossetine in the CV-induced culture by HPLC mass at the time when the chromosome-free state of the E. coli strain was confirmed. However, it is possible that this production was due to the detection of crossetin produced by E. coli before the chromosome-free state or by an escape mutant. We were the first to propose the concept of using restriction enzymes as described above in iGEM, and we were also the first to successfully demonstrate chromosome-free in E. coli. Our system has strong potential, is highly standardized, and can be adapted to most bacteria, and is expected to be used by various iGEM teams in the future. The mechanism of action of our system and the restriction enzyme sequence used in this process are available on the parts page. Finally, the antidepressant effects of crossetin, crocin, and pecrocrocin have been reported on a clinical scale, but their specific mechanisms of action have not been elucidated. Therefore, we used simulations to predict candidate mechanisms of action for these three substances. Although the cause of depression is not completely clear, reuptake of norepinephrine is thought to be one of the solutions. Previous studies have suggested that the three substances may inhibit norepinephrine reuptake, and we specifically targeted the norepinephrine transporter, NET, which is known to be involved in the reuptake. We performed docking simulations to predict whether these three substances could interact with the binding site of norepinephrine. The results suggest that crossetin may not bind to the binding site due to its large distance from the site. Interestingly, the possibility that glucose may interact with crocin, which is a glycosylated form of crossetin, emerged. Picrocrocin was also suggested to interact with NET by a safranal molecule. Based on the above, we did not fully elucidate the function of crossetin, but we were able to predict its mechanism of action to a large extent because there were suggestions of potential interactions with NET for crocin and picrocrocin. For social implementation, we partnered with a local patisserie and a Japanese saffron producer to create prototype cookies infused with natural saffron for a social initiative. We gathered feedback from depression sufferers and their relatives. We conducted a SWOT analysis, created a lean canvas, and prepared a financial plan. Market research and conversations with depressed patients confirmed our project's viability. Furthermore, to ensure the acceptance and effective implementation of our project, which involved genetically modified organisms, we prioritized inclusive learning. We employed diverse educational methods, including classes at various educational levels, online platforms, contributions to libraries, online events, and laboratory sessions to make information about synthetic biology accessible to people from all backgrounds. Additionally, all educational materials, such as slides and protocols, are publicly available for anyone to replicate. 320 million people, which is almost the same as the population of the United States, actually represents the number of people worldwide to whom we can offer our helping hand. Just as vitamin C helps invigorate our body, the time for achieving mental health through simpler means is almost here.